I have the pleasure to introduce our first speaker this morning, Dr. Dana Stanley. She's a professor at Central Queensland University in Rockhampton, Australia. Dr. Stanley was awarded a PhD in molecular biology from Victoria University in 2009. Following graduation, she started a postdoctoral position at Cicero's Animal Health Laboratories in poultry intestinal health research. Dr. Stanley specializes in gut microbiota and genetics during her time at the Animal Health Laboratories. In 2013, she moved to Central Queensland University and was recently, she became a professor there. She is now the leader of the Molecular Biology and Poultry Health Research Cluster in the Institute for Future Farming Systems, where they focus on human and livestock intestinal health and nutrition, probiotics, next-generation antibiotic development, and pathogen control. Dr. Stanley is also investigating ways to control and reduce antimicrobial resistance on farms. Recently, she started working on pig microbiota and has several projects running directly on farm under commercial conditions. Dr. Stanley is very passionate about nature conservation, animal ethics, exercise, martial arts, and nutrition. Please help me in welcoming our first speaker this morning, Dr. Dana Stanley.
Hi everyone, my name is Dana Stanley. I come from Central Queensland University, Australia, and I'm going to talk about livestock microbiota manipulation. I want to thank the organizers for inviting me to this conference. I was quite surprised because most of my work in gut microbiota and intestinal health is in chickens and in humans. I just recently started working in pigs, and I found that quite challenging, to be honest. I was picking the phone many times asking my collaborators, what is parity? What is this? What is that? But I'm getting there. My presentation today is going to start by overview of methodology limitations so that we can learn to better understand the data and interpret the data. And then I'm going to talk about different ways of manipulating gut microbiota and specificity of chickens and pigs as well, and a little comparison between the two hosts. So during the last decade, quite a few tools to investigate microbial community became widely available. We have a lot of omics. Multi-omics is a buzzword at the moment. This spiked growth in investigation of microbial communities, and new knowledge is really rapidly accumulated. On the other hand, there is lots of confusion. There is lots of misinterpretation, and I have to stress there is a lot of data misinterpretation, overestimation of what these results are showing present in publications because the methodology just skyrocketed very fast, and but a microbiota requires very strong bioinformatics background to understand really how to interpret the outputs of this analysis. Two main methodologies exist for microbiota investigation. We have 16S amplicon sequencing, and then we have metagenomics. And although they're fundamentally really different, they're often interchanged incorrectly. So I'm going to start with overview of these main investigation tools, about pros and cons, about issues with the analysis, and then into real data. So microbiota is amplicon sequencing, refers to amplicon sequencing and investigates taxonomy, answers the question, who are they? So if we have mock community and we sequence microbiota, we will sequence one small section of the DNA based on primers chosen. We will amplify just 16S ribosomal RNA gene, and based on that marker gene, we will then, after sequencing, do the analysis and figure out the taxonomy of each one of these amplicon sequences. Microbiome or metagenome investigates total DNA. So there is no amplifications, there's no PCR, there is no short fragments and just one amplicon amplified. We take all of the DNA, we fragment it into smaller pieces, and we sequence the whole lot. And then we take smaller pieces, we put them together, and then based on those contigs that we assemble, we estimate taxa, genes, then we can look into all pathways present in this community, what this community can do, what functions it has. We can also blast it against resistome databases and get resistome profile, which is list of all antibiotic resistance genes that we have and their abundance in the community or each individual animal. We can also look at viriome if we look into databases that contain viruses. We can assemble metagenomes. It's much more data, much more reliable, and much more expensive, and not yet close to outcompete microbiota or classic 16S. So this is a future of the area, and I'm going to touch this why as we go. And this is the presence, the 16S, this is our presence at the moment. So main issue here is PCR bias. The PCR bias is even more amplified with the fact that we didn't choose the best marker for this type of analysis. So, but we have a very long molecule, about 1,500, for example, nucleotides, but we can amplify a very small section of it. So we have to pick which parts we amplify. And we are obviously going to pick variable regions that give us the most information, but different variable regions give us different information. We can see that there are papers picking some of the variable regions as better than the others, again, it's very difficult to compare or to analyze together data sets from both of these methodologies. So what we are doing with each variable region, even if we just change one nucleotide in primer data set, instead of analyzing the complete community, we are taking one subset, like a handful of it. There is no primer set that will pick up all of it. Only metagenomic sequencing will pick up all of it. 16S will pick up whatever primers can pick up. Change one nucleotide in forward primer and you will pick up a little bit different community again. If you change variable regions, again, you're picking up something, another handful, but again, representative subsample of the community. This is something to have in mind. The other thing is, as I said before, 16S was a bit misfortunate choice for marker. Later on, we discovered that different species have different number of 16S ribosomal RNA genes. So different number of copies of this gene that we're amplifying. So unfortunately, say some other candidates like REC-A and RPOB have one copy, so one sequence would theoretically equal one cell. Here we have different copy number, and this paper goes into details of it. Some species have like 15 copy numbers in this particular subset that they analyzed, and others will have one, but most of them have more than just one copy. So this is what we get when we look into microbiota analysis. We get OTU table, we get these comparisons of microbial communities, and these comparisons, these ordination plots and these bar chart plots that can kind of tell us what we are comparing can be really useful. But we have to be very careful when we decide to call the taxonomy or to call certain OTU by species level. So we currently facing a big sort of dilemma in the community. There's a big discussion. Should we use OTUs or ASVs? Now, ASVs are proxy to isolate level. OTUs are proxy to species level. ASVs, when you take all the sequences from amplicon sequences, and all of the 100% identical are together and they make one ASV. OTUs say that we have between 3%, for example, difference is allowed within the same species, and we pick the species with allowed 3% divergence between the sequence identity. So Important thing is to know that neither is correct. Both are approximation. Neither is giving you, ISV will not give you exactly isolate level. OTU will not give you exactly species level. OTU will overestimate richness, and denoising, which is a method in dealing with microbiota, can often underestimate richness in some environments. So OTU tables and ASV tables that you see in papers that are coming right now, so the latest, we're switching from kind of OTUs to ASVs, they give comparable, but they give still different results. And here is an example. In this particular table, each bar is different pure isolates. So we took a pure strain that we isolated from chicken guts in this case, and we sequenced it. And this is ASV denoised data, data to denoised data. And what we can see here is that, for example, this particular pure strain, pure isolate, is split into 2 ASVs. Some of them are split into 3. And if you go here further, some of these ASVs will, this one is split into 2, and so on. So there, not one of these methods is exactly correct. And in some cases, for example, here we obviously have 2 copies of 16S and 1 copy aligns as is identified as this particular one isolate and the other one is another. In some cases, these isolates can, these 2 copies from the same pure isolate can actually be even aligned to different genus. So we need to be very careful when we're calling something species. And here we are in the last slide that touches on these issues in the analysis that helps us analyze the data, actually interpret the data a bit better. Silver or green genes. We have 2 databases. Green gene is very popular. All the papers in pigs that you read will probably be on green genes unless in last few years, because green genes were the sort of format of green gene output. Green genes output is compatible to a lot of other softwares, and that's how it went. So however, green genes is not updated for like 10 years, and culturomics exploded just like microbiota, and so many new species are discovered, new genera, new families. And all of these things have been updated in SILFA database. This is just one example. This is ASV number, and this first column here is green genes taxonomy for the same ASV, same sequence was submitted to green genes and to SILVA. And what we're seeing here, we have different families here are different. In this one, we see different genus for the same, obviously same sequence. Here in green genes, we have a lot of, look, there is hundreds and hundreds of sequences that will classify in green genes just as bacteria. Whereas in SILVA, they are taxonomically identified even to species level. And I can go on on these things where we have different, see, this is a different order even in between the 2 databases. A family S247 does not exist anymore, it's reclassified. But the major differences are in Clostridia. Clostridia have been split to Fastidiosphilia, then Lachnoclostridia, Eryspiloclostridia. A lot of different genera came from what we used to call Clostridium and still call it Clostridium when we do taxonomy through Green Genes database. And so we touched on all the issues in microbiota analysis. Functional analysis can give us a lot more, as I said before. I'm not going to stay too much on it. About things we can get from this analysis, but instead of OTU number, we get the same output, similar output. In this case, obviously, it's normalized output. But what we're getting pathways or functions or antibiotic resistance genes or viruses or whatnot, instead of taxonomy that we had in previous data. It is important also to know that Human Microbiome Project and Human Microbiome is one of the most expensive consortia that we've seen in science, discovered that microbiota is extremely variable. So between your sample taken between today and tomorrow and this evening will be slightly different. On the other hand, these functions, the functions that I was talking about here in full metagenomic analysis, functions are stable. So we just have different bacteria in our gut that do the same thing. They do the same thing across the globe, across the continents, but using different tools or different taxonomy. So now we go back to agriculture, and this was like 13 minutes introduction into the methodology for those of you that are not very familiar with microbiota analysis and issues in the area. So how do we use this knowledge in agriculture? I don't want to dishearten you. I don't want you to think that it's a terrible method. Look how many issues. Every method we can pull apart and find so many issues. Fact is, microbiota has given us a lot of therapeuticals, a lot of diagnostics tools, and it caused the biggest growth in businesses in last decade. Like, how many businesses you know that where you can take your sample and get microbiota analysis, and plus those that actually cater for industry and agriculture. So it does work. We just need to back off when we interpret the results. So we are seeing big differences between chicken, pig, and ruminants, for example, when we look at livestock gut, especially ruminants. On the other hand, chickens and pigs kind of bin together as opposed to other livestock. They share many common challenges that if you look into genus level of pathogens, they share pretty much same genera on major problems like Escherichia or Salmonella or whatnot. We have products and supplements that are very similar. So products like phytogens, probiotics, a lot of these products are catered, the same product is catered for pig and for chicken. And we can actually look at data that comes from chicken and pigs interchangeably, because if certain phytogenic sort of antibiotic alternative controls or completely removes, I don't know, Escherichia, for example, it will do the same most likely in different communities, whether they come from pig or chicken. And we also have some major challenges in both industries. Could be control of pathogens, so to run the shed disease-free, and AGP removal climate. We have to adapt to not being able to use antibiotics in industry anymore. So, and that opened a lot of issues. And again, a major issue there that suffers is control of pathogens, pathogen load. We have a lot of animals in small space, literally walking on their own excreta that is loaded with pathogens, environment that is extremely pathogen rich. The issue in chickens also now with free range is, if you ask farmers, it's a disaster, because in the barn or in cage they could easily do biosecurity, and disease is still very low in these systems, but open range and free range, it's a disease show for the farmers. So what we're trying to do in agriculture using microbiota analysis and microbiota manipulation is trying to achieve concept of controlled colonization. This means colonize animals from the get-go, from day one, with good microbiota, let good bacteria colonize the gut and take over, and then they will outcompete any hopefully new coming pathogens, or at least fight them and prevent colonization of bad bacteria. This is easier said than done, and I am going to take you through a few lessons that we learned in trying to achieve controlled colonization in chicken. With chicken, we have a lot of problems that pigs don't have. In chicken industry, we have poor reproducibility between flocks. And the answer to why we have the same flock, and this is the paper that shows three trials, but in the same facility, same batch of feed, and then buying 3 batches of chicks from the same hatchery, from the same parent stock. And one of these trials was dominated with Firmicutes, and the other one with Proteobacteria, and another one with Bacteroides. So completely different, extreme different in community richness, in community diversity, in what is colonized these birds. So why? What we realized during this experiment is that chickens are removed from maternal microbiota. So the eggs are taken from the breeding stock, hatched under fumigated, almost sterile conditions, almost sterile, like they disinfect and fumigate everything, and everyone is wearing gloves and they're fully suited when handling these chicks. So there is no mom and there is no microbiota that they get from mother. Maternal influence is gone, and these birds are colonized with truck bacteria, cardboard boxes bacteria, plastic boxes bacteria, depending on how they transport it, and so on. Again, we also have issues with layers and broilers, because obviously broilers grow for 6 weeks tops and then they're killed, and so they're killed in very young age, whereas layers, they'll reach maturity. Early microbiota and mature microbiota are very different. Broilers, 6 weeks old broiler, you cannot literally hold it in your hand. 6 weeks later, it looks like a little chickie, like a week-old broiler. So when you give them food, broilers will be dying eating. You have to turn the lights at night, turn the lights off to prevent them from eating themselves to death. Layers will just look at it and play and lie down and chill. So difference is very big, not just in microbiota, but difference is how these products work, the probiotics, phytogens, pathogen control, because there is massive difference in feed intake, so doses will be different. And then we're comparing young, we're comparing birds that don't have massive influence of hormones in younger birds when we're with all female layers. So it's almost like having two species that are not comparable to each other, as opposed to piglets where the situation is much more uniform. So how do we achieve controlled colonizations? What do we do with chickens? So we try to get healthy bacteria into the hatchery from the day one, so that obviously we have to remove maternal microbiota, and the eggs will be fumigated because no one wants to bring salmonella or something, so they want hatcheries to be super clean. That is not going to change. But can we give them probiotics immediately post hatch, something that will come in and line up the gut? Can we then allow that maturation of microbiota happens without pathogens by controlling what is in the feed, what is in the shed, and what rodents or birds have access to them? And I've been to piggeries in my latest moving into pig research adventure, and I have seen birds eat from the pig, because in Australia we don't, in many cases we don't even have super fully enclosed facilities. Some of them are just open sheds, and the birds can get in. You get snakes, we get rats, we get all sorts of things. So how do we do it? How do we control it? Why do we need to control food and nutrition? Nutrition is a completely different story, but the whole food is always part of microbiota studies. We always say this was the recipe, or this is the magic sort of feed formula we used to grow these animals in the industry. In a lot of nutritional sort of interventions, we see the effect of, I don't know, soy meal, or the effect of barley, the effect of whatever on gut microbiota. But we look at what this particular food ingredient will do in the gut, which bacteria will increase, which bacteria will decrease. No one was ever looking, is there any bacteria in the feed? When I went through all of that data, I looked through a lot of things on Google Scholar, on PubMed and whatnot. I didn't see any papers on looking what is in the feed. All the papers were saying what the feed does to existing microbiota if you change the diet. So any nutritious environment will have inside bacteria that live on it. Even our liver and our organs are colonized, and this is now, it's something that is hard for us to accept. Our gut leaks all the time, and our organs have their own microbiota. So if we look into interesting examples about it, gut bacteria in Japanese people has sushi digesting genes. They can literally digest seaweed, and that bacteria actually came because they eat a lot of oysters and a lot of seafood, and that those, by eating that food, they colonize themselves with ocean bacteria. Interesting is that Japanese guts are abundant with bacteria that are coming from rice, from miso soup, pickles, seaweeds, fish, green tea, meat, and coffee. And they have very different intestinal microbiota. And of course, they live much longer, and they have very healthy lifestyle. Not much obesity among Japanese. If we look into feed, then what is coming in the feed, we were very lucky to be friends with one of the major feed companies in Australia, and they sent us the data from different mills that supply, 4 different mills that supply raw ingredients to this feed packaging company, and it all came with all the information about longitude, latitude, where it was collected from, like the whole history of each one of these, of the samples in each one of these bags. We then sequenced microbiota and wanted to see, is there any bacteria in it? A lot of samples were also, there was no DNA in them, but in a lot of samples we also had quite a lot of bacteria. Most of the samples had a lot of bacteria in it, and here's the similarity of different sources. So meat and bone meal, blood meal were very different. Poultry oil also to a certain extent, and all here you can see also all of the grains like barley, canola, corn, mill run, oats, sorghum, even soybean and wheat, they all overlap. They have similar carb-loving and fiber-loving bacteria. But again, we have here we have blood meal and meat and bone meal. Limestone were little rocks that they put into chicken feed to help digestion, because of the way they always, like in nature, they will always swallow little rocks to help the gizzard crunch the food. So that's added into feed. This is probably not added into pig feed. I wouldn't think so. But we saw there's different communities. We looked into their richness, and the highest number of different species that we could find identified using the methodology and analysis pipeline that we used. The biggest highest richness was in blood meal and meat and bone meal, and in poultry oil to some extent. So the next thing that I wanted to stress out, that we noticed that when we put the samples based on supplier, on the mill from which the company supplied these feeds, and we had we had few bags from each different mill. So a feed that came from suppliers was matching the sample to sample similarity and group to group similarity graph was matching the geographical locations. So supplier A was a bit different because it's coastal and warm climate here, although it doesn't look that much different in on the map. It's a bit colder, and these two overlap. They service this region, whereas this supplier is servicing probably that region. And here we have Brisbane, which is tropical, subtropical climate, and we do have different bacteria and same feed ingredients, just geographical location. There is lots of other things that would influence which bacteria grow in wheat from tropical or continental, or again coastal climate. When we sequenced we fed the chickens with that feed, and we followed the feed through different gut sections to see and sequence the gut section's microbiota to look into what happens with that feed when it gets into the animal. We noticed there's only 6 genera that were just found. Oops, that were just found in the feed and not in birds or anywhere in the gut. Most of that was from limestone. If we look here in Ilium, for example, there's quite a few genera that are shared. If we look at genera shared in the Cecum, again, there's quite a few. So most of these genera are shared. We see that feed has the lowest number, this is different feed. This is finished feed from different supplier, different experiment. We could see that this finished feed, had different, obviously lower number than all of the gut sections, but nonetheless, it looks like it's possible that it seeded the guts of these birds. Possible, but we cannot be sure. And we cannot be sure because during the pelletizing process, not everything is killed. It's a snap sort of snap heat for a very short amount of time, and a lot of species survive. So you will remove maybe 90%. I'm just shooting this from top of my head, of bacteria, but those ones that are left, they are, and we can detect live bacteria in every feed. Even if it's pelletized, if we try to grow it, we'll grow a lot of different things. And those things will, in chickens, sit in a crop and just bloom while they're waiting to get into the gut. So the other option exists that this food colonized the birds, but the other option is that that we could be measuring dead bacteria from the feed. And what does that mean? It means that methodology picks the DNA and amplifies, doesn't care whether DNA came from live or dead bacteria. On the other hand, there is lots of papers that say that yes, you can detect dead bacteria in upper gut, but after proventriculus, where we have pH of like 2, and all these digestion is happening, DNA, all dead DNA will be degraded. There is lots of evidence of that. So that means that cecum and ilium and excreta should probably have these species, or these genera that are detected, should actually be live, and the fact they overlap with those we found in the feed doesn't necessarily mean that they come from the feed, but we also checked OTU level and we're getting the same OTUs, which would make it very unlikely that they somehow miraculously existed in our shed and in the feed that we purchased for this particular experiment. I have to say that sheds are fumigated and steamed between the trials, and they are experimental sheds. So what we're talking about here is that just like Japanese guts get seeded with bacteria from ocean and miso soup and whatnot, our pigs and chickens will definitely be seeded with the bacteria that come in the feed. So feed needs to be controlled. Here is another experiment we did, and in this experiment we had quails that we put into germ-free lab, and in that germ-free lab we actually split the birds into 2 groups. One group received just sterile saline solution. The other group, immediately post-hatch, received Lactobacillus agilis. Then we had them in clean room, sterile feed and whatnot for a week. And after that 1 week, we placed them in the room in shed with other quails. And they spent, so from week 1 to week 4, they spent that time with other quails, co-housing. And what we saw is they never recovered richness. Their richness index, they had like 20 to 30, 40 OTUs, these ones that were sterile controls, and these ones, the ones that were given lactobacillus, some of them would have like 10 different species in their gut, in their, like they never were able to reach the richness. So once they were colonized, that was it. Not much was able to colonize after. The amount of lactobacillus in those that received controlled sterile saline solution was 0. There was 0 lactobacillus in ilium or cecum of those birds. And this is how their guts looked. Didn't matter after 1 week that we placed them with other birds. They couldn't get one lactobacillus species to colonize them. And the ones that were colonized and other things were prevented from colonizing, those birds had a lot of lactobacillus does not belong in cecum, so it's over-colonized these birds. So the point is, it's possible to manipulate. We did lots of other experiments trying to manipulate microbiota. So can we design a microbiota by giving the birds now something more manageable? Just give them a little bit of small sort of live probiotic strain immediately after they hatch and put them in a shed with other birds. We did this with different lactobacillus strains, and there were few strains out of many, only few worked. Lactobacillus johnsoni, Kitastonis, and plantarum-like strains were able to protect birds from leaky gut challenge. And in leaky gut challenge, protected birds showed that they have much higher amount of Treg cells and much better ability to control inflammation. And those birds were not getting diarrhea and putrid litter like control birds. But when we looked a bit further and deeper into well, you get very excited. We discovered an amazing way to colonize the birds. Some of these actual big increases in Tregs had side effects and negative effects. And this is why we need mechanistic studies to have a look at what's happening. So out of probiotics that worked for us, only one had 0 side effects and was really amazing. On the other hand, things that worked in broilers didn't work in layers. Things that worked, probiotics that could colonize and protect in male, didn't work in females, and vice versa. It is a very complex issue, and there's lots more work to be done in trying to control the colonization of the animals using many different ways, some of which I've covered now. What we need to do in pig industry, from the chicken researcher perspective, we need to make sure that little piglets are colonized with very good microbiota from the day one, which means, if possible, enrich immediate environment with good bacteria, with probiotics, to exclude or outcompete the bad bacteria, maintain good maternal microbiota, so look after sows' guts, and be very careful on products that are given in gestation and lactation. Competitive exclusion is a good option. Spray beneficial biofilm products, for example, microbial biofilms of beneficial strains that can be sprayed on a wall. They actually do competitively exclude pathogens a lot. Fresh feed, make sure that feed is properly stored so we don't have open bags that are gone moldy and so on. Look after the feed, because feed carries a lot of bad stuff and that can actually get into the guts of your animals. Rodent bird control, biosecurity. Each product that is used to control pathogens has different mode of action, so they could be all 5 different phytogens. They will all have different benefits, target different microorganisms, and have different side effects if they have any. Use the data between pig, chicken, even human data, if microbiota, if you're interested in gut health of your animals. Don't restrict your reading only to pig microbiota and pig gut health. Each product will change gene expression, and we need to be aware of that all of these products that we give, probiotics, supplements, different feed formulations, have low but subtle influence on organ health, weight gain, animal behavior, disease predisposition, and including, but not just, gut health. So regardless that these products are coming into the gut, they can influence unrelated systems of the body. Now, on behalf of my team at CQU, I want to thank you again for allowing me to talk today. My team there's 8 of us at the moment, and 3 new members are coming early 2022, and we are very much interested in work that is happening in pig health and pig microbiota. Thank you again for the opportunity to talk to you today, and all the best.
All right, and I believe Dr. Stanley is joining us. Can you hear us okay, Dr. Stanley?
Yes, I got tired from listening to my own voice.
Okay, great. So we'll have the speaker boxes like usual for any questions.
Dr. Stanley, thank you for that. That was very interesting. As we think about the microbiota and manipulating the microbiota, knowing that microbiota populations are different from animal to animal, as well as from herd to herd, when we look at future research with microbiota manipulation, with probiotics, prebiotics, functional feed ingredients, in addition to characterizing the microbiota or the microbes in feed ingredients, do you think there's a need in the future to develop techniques where we balance the microbiota in the research animals across treatments to avoid some of that bias that might occur?
Well, thank you for your question. There are many attempts to kind of standardize microbiota. There are products on the market that resemble, like, at least for chicken, that resemble like a ceco transplant. But a lot of these products, there are a lot of companies that are trying to achieve that, but no one has really completed that task. Because whatever you try to do to inoculate the chickens, a lot of other external influences will come at play. So in my opinion, if people want to standardize the microbiota, everything else would have to be standardized, like feed, and every little feed ingredient will fluctuate the things. There is a little bit of difference in genetics. Then there is also colonization. So, I mean, if I was given tasks to standardize microbiota, I would think of it as mission impossible. It was something ideal, which would be great, but I don't think in current conditions. And if you go through the farms and you see how different production systems are and everything, different people handling a different climate, different temperature, you change one little tiny thing and gut responds immediately. So I think this is the reason why many companies are now focusing on controlling function and ignore taxonomy completely. And this is where this area is now moving towards, yes, metagenomic analysis is much, much more expensive, but the prices are coming down. A lot of people are opting for shallow sequencing, so to pay a little bit cheaper. I don't need 100 million sequences. Maybe I can get some information from 10. The only issue that prevents that progress is the price. Sorry, I hear a lot of background noise, so I don't know if you can hear me okay. So a lot of companies are moving now towards when they're developing new product, they're not looking into taxonomy. So they're not looking, did we remove E. coli, did we remove Campylobacter? But instead they're trying to check if they're removing pathogen functions like toxin production or invasion of epithelial cells involved genes. So they look at enzymes and genes in community rather than trying to do taxonomy, because based on my talk from before, we can't ever really know taxonomy correctly anyway. I hope it's a very long answer, but I hope it kind of answers the question.
So I have a question about application. So we know when we look at these responses that we can change microbiota, right? So, but it's hard to interpret that and where we go with that. One question is, have you seen changes in animal performance where you do not see a change in microbiota?
So if you sneeze, your microbiota will change. So whatever we compare, we always have a paper, because there will always be this O2 is up and this O2 is down. This is why it's such a profitable area for paper publishing. So I've never seen that change in FCR without changing in microbiota, but I have seen microbiota changes, big changes that don't really influence FCR. So again, it can be manipulated, and there are a lot of products and shifts that can be targeted. But again, things that change FCR are very often not reproducible across different sheds, in my opinion, in my chicken-based opinion. I only have done like 3 trials on pigs so far.
Dr. Stanley, it's John Patience here. Thank you so
Hi,
much
John.
thank you so much for your presentation. You just nailed it, because we wanted to get a better understanding of the microbiome. I think the microbiology community has done a really good job of convincing us the importance of the microbiome, and so we know it's important, and we don't always understand exactly what we should be looking at or looking for, and I think you've done a really nice job of that, so thanks very much. And also it's become so much cheaper, right? It's so much cheaper to do the work than it used to be, even within the last 5 years. So my question then is, what do you see as the next big thing in the microbiology world, the world that you live in, as it would affect the world we live in, animal science? What do you think is coming down the pipe that's really going to change things in a big way? Thank you.
Thank you. Thanks for the question, John. So what I think is happening at the moment, there is a little shift from microbiota to microbiome, and from taxonomy to function, that not many can follow. So you need to be really well funded to follow the trends in fashion in this area. But the other thing is, there are a lot of transcriptomics and metatranscriptomics are also coming in. Transcriptomics is the step that relates now this bacteria with the host. So if we do RNA-seq of ileum of the piglets, and this is, I'm very excited that I'm doing this sort of pig transcriptomics now, that what we can see is, for example, any nutritional change or microbiota change will relate to turning on and off certain genes, or modifying expression of the host. But now we have so much more machine learning coming into this bioinformatics, and multi-omics methods and systems biology methods to analyze the data, so that we can actually try to correlate or connect a change in certain taxonomy to change in function, and then follow that to change in this particular enzyme, and then link that to genes that are influenced in the gut of the animal. So trying to see a bigger picture, I think, will require more money, more bioinformatics expertise, people that can do machine learning, which means you need to hunt your postdocs from different areas if you want to be in it, because it's very difficult, like to find a normal postdoc and tell them, go learn this. Like, it's not going to happen. But it's important also to remember that novel algorithms and machine learning allow us to predict things with very good certainty. Like, from transcriptomics and microbiota, we can do disease profiling. We can predict what treatment can amplify the effects of, say, our treatment, what treatment will undo the effects of our treatment. We can, without running 2 treatments together, we can actually see and estimate to very good level how they're going to influence each other. Are they going to work well in combination or not? And I think those things are maybe 10 years down the track till they see the real peak. But this, the way I see these, and from the amount of time I've spent doing research, I can see that microarrays were such a big thing, massive fashion. They go up and they go down. Microbiota will also go down, and it is on down trajectory. But new things are coming up, and I think it is functional analysis, transcriptomics. Metabolomics is the science that teaches us how they speak to each other. Like, the language of bacteria in our gut is metabolites, and like in Star Trek, universal translator is your LCMSMS. And that is now, again, people are finding out how they communicate, how they actually talk to each other and plan how to attack the host together. A lot of amazing papers are coming in future, I think.
Okay, great. And for the sake of time, I think we'll go ahead and thank our speaker one last time.
Thank you. Thanks again for inviting me.